Good morning. My name is Ed Conway, and I'm the director of the Center for Blood Research, and it's my pleasure to welcome you today to this 10th Earle Davie Symposium. So before we start, I do have some business items. Now, I know we have many Americans who are visiting up here in Canada. Now, I don't want to be political. I do want to point out, this is a gift of Alyssa, that this is not always the case. We do have some nice views to be seen here, particularly in the summer, but also in the winter. Now, if you decide to stay a little longer, and I'm not encouraging you to do anything one way or the other, but this is the means of getting Canadian citizenship. All you have to do is memorize the parts of the Mounties uniform, and a 50% is a pass rate. All right, now on to real business. First, there are washrooms on the main floor and downstairs. There'll be 2 coffee breaks. Buffet lunch, stay until the end. There's wine and beer and some food to be had. There's the posters should all be up. Judges to judge during the first coffee break and through at lunch, so ideally finish before the end of lunch and hand the results to Hannah and Anna. There will be online evaluation forms emailed to you, I think, so please complete those because we really do appreciate feedback. CME credits for those who need them and haven't registered for that, please let somebody at the registration desk know that as well. Feedback. So most of you know about the Center for Blood Research, but those who are not aware, the CBR was conceived in 2012 by 5 scientists at University of British Columbia. That was Don Brooks, who is here, Dana Devine, Grant Mock, and Chip Haynes, and led by Ross McGilvery, was our founding director. And Ross, wave, there's Ross. So he's the initiator of this very special center. It's quite unique, actually, in Canada and in North America, in that it was built on the concept at the time, which was new, a multidisciplinary research institute devoted to blood research. Indeed, it is multidisciplinary. We have 6 faculties represented. It is now 40 investigators and clinicians throughout UBC, with a headquarters at the medical school on UBC campus. We particularly promote education programs and research interactions, or translational research interactions between the docs and the PhD scientists, and we believe that we're doing really very well at that. Now, this doesn't happen without some costs, so we do a lot of, it's a lean administrative team. That's Ed Prydell, who's the associate director, Hannah Kim, who many of you know, Anna Sinova, and Amar Grivel, who are really responsible for putting this event on and all our other events. As I said, this doesn't come without a cost. We really rely entirely on donors, and here you can see our major donors for this particular symposium, but we also count on donations from you. So if you want to donate towards a summer student, a fellowship, or buy a building, just come and talk to me, and I'll be happy to chat with you. So this is why we're here yet again, our 10th time. It's just over 50 years, actually 52 years in a couple of months, that Earle and Oscar Ratnoff described the coagulation cascade. And you can see here, this is from the first publication when it was in 1964. Now, a little history about Earle, and some of you know this story. But Earle grew up in Tacoma, Washington. This is the public school where he went to. And from a very early age, it was evident that he was particularly interested in building things and in clotting systems. And he was also interested in sailing. from an early age. And he obviously did very, very well, and didn't take long. He looks a little, just a few years before he ended up here at the University of Washington with Hans Neurath doing his PhD studies, where he was encouraged to look at how does trypsin get activated. So how does it go from its precursor to its active form? And really this was the first step towards the notion of limited proteolysis, which is obviously crucial in pretty well everything we do as scientists and clinicians to understand the mechanisms of action. He later on then moved via Boston and then ended up at Case Western, where he worked with Oscar Ratnoff, and that's where he first described factor 12 deficiency, and which is known as Higman factor at that time, and then subsequently, which really set the stage for this, which we know it today, and that's not as complex as it is. But he subsequently cloned and purified and characterized the structure and the function of multiple coagulation proteins, and you can see just some of them here. So it's quite remarkable when you think about it. Pretty well everything we work on, he started. So that's why we're here. And it's not just because he was a great scientist, but this has had a huge impact in the clinics in really every discipline. So every PT and PTT you do is based really on these discoveries. So we're here to celebrate that and to recognize that. And the impact that he's had over the years is felt right here because we have this symposium and we transmit sort of our new ideas and concepts and discuss them further so we can make advances, hopefully a little bit of advances like he has. So what we're going to, this symposium, just by the way, was initiated 10 years ago because Ross McGilvery was a postdoc in Earle's lab and started this as an idea to bring Earle up from Seattle and have some keynote speakers and gather people like you together, and it's been an extraordinarily successful venture. We're really pleased to have Earle here come each time. He's given talks before. Maybe he will give a little chat at the very end. He brings a good friend of his, Eddie Fisher, who gave a Nobel laureate who gave a talk a few years ago. You want to hear great stories? Eddie, as he says, knows Earle for about 120 years, and he can give you some great stories of his younger days. So we're going to start into the program. So our first speaker, once I get it set up. So our first speaker and keynote is Dr. Nigel Key, who is an adult hematologist specializing in non-malignant hematology. He particularly affecting coagulation, and he really dances on both sides of the system, on the bleeding side as well as on the clotting side. Nigel is a Harold Roberts Distinguished Professor in the Department of Medicine and Pathology and Lab Medicine at University of North Carolina at Chapel Hill. And many of you know Harold Roberts. He was a keynote speaker here several years ago. Nigel also is the chief of hematology at UNC Chapel Hill and carries several other hats as well. And he's the past president of the International Society of Thrombosis and Hemostasis, so he certainly gives back in a big way. He hails really from Zimbabwe, but he grew up in Ireland and then did his training at St. Andrews University and the University of Manchester, and then did further training in internal medicine and hematology in Bath and in Oxford. Then he spent about 15 years in the University of Minnesota, and then finally was recruited to North Carolina just over 10 years ago, where he leads a very, very active clinical as well as basic science programs. He's extraordinarily well known, and mostly not only for his great science, but because he really does give to the community, to the research community, to the clinical community, and it's really a delight to have him here. His publications are many. So I'm going to ask Nigel to come up here and give the first talk, which is really a great way to start the day, considering it's a factor 12 talk, which is really where Earle started.
Well, thank you very much. I'm really delighted and honored to be invited, especially as a keynote speaker, being a key myself. So, and you've seen this before already once today, so I'm really kind of intimidated to come to the symposium and talk about factor 12 when I have such a distinguished biochemist and scientist sitting in front of me, actually many of them, but this is the original cascade or waterfall model of blood coagulation that Earle and Oscar Ratanoff proposed. And actually, most of my efforts over the years have been more focused on the tissue factor pathway, so the factor 12 is sort of new. And I feel like bringing factor 12 here to this symposium. Where I come from, they talk about carrying coals to Newcastle here. It's basically otherwise called selling sand to the Arabs or ice to the Eskimos. But anyway, I'll give it a go. So this is probably a more contemporary view of where coagulation stands. This is from David Galani's group. And a few things to note here that we all accept that on the left-hand side here, oh, I got it, okay. Yeah, okay. So on the left-hand side here, that this is all to do about thrombin generation, that this is the tissue factor pathway that ultimately generates thrombin. And then we have factor 12 sort of hanging out here with the other contact factors, high molecular weight kininogen, prekallikrein, with the ultimate goal of generating not only factor 12A, but also some vasoactive kinins and has multiple effects that go beyond coagulation. So what was, over the years, factor 12 and the contact system was sort of put on the back shelf, I would say, until recently. And there's a lot of interest again now for students who are not in this field about really targeting the factor 12 as a potentially safe anticoagulant. So this begs the question, what is the role of factor 12 in hemostasis and how does it get activated? And more importantly, I'm not going to get into this, but how does factor 12 decide what to do? Usually when it gets activated, it actually leads to a syndrome known as hereditary angioedema, and it's a major target in that situation that really has very little, if anything, to do with thrombosis. One thing you see is that when factor 12A is generated, it can activate factor 11. And then subsequently, David Galani and George Brose had shown that thrombin also is a second way of activating factor 11. So factor 11 sits here at the junction between the contact system and the thrombin generation pathway over here. And one of the issues becomes, when Earle did his work and others showing that when we do an APTT test that we can activate factor 12 through artificial negatively charged surfaces. And one of the issues becomes, if this is important in vivo, what is the surface or what is the activator of factor 12? And part of the resurgence and the interest in factor 12 has really come about from the recognition that platelet polyphosphate, which is just repeating phosphate units, the platelets dump into the circulation when they're activated in high concentration, can be one of the mechanisms for activating the contact system. So another proposal is that, separate from polyphosphate, that these entities, neutrophil traps or extracellular traps, nets, can actually activate the contact system. And this is what I'm going to talk about mostly today. So just to remind you, I see the emphasis also in infection and inflammation in the Blood Research Center, that neutrophils can kill bugs through traditional ways of both protease and reactive oxygen species, but more recently the recognition that these neutrophil nets, which occur, this is sort of summarized here very briefly, that there's nuclear translocation of the granular enzymes such as elastase and myeloperoxidase, as breakdown of the membrane, chromatin decondensation, and then this aptly named net formation where the neutrophil throws out this chromatin with a backbone, of course, of nucleosomes and decorated with various enzymes such as elastase and myeloperoxidase. And this traps bacteria but concentrates bacteria, and this clearly is an important mechanism for bacterial killing. But and this was the original publication that really set the field alight, which was from Brinkman's group in 2004, front page of Science, showing that these nets, and you see this sort of beautiful structure here of chromatin, are decorated with neutrophil elastase, myeloperoxidase, and the nucleosome units, the chromatin units, consist of DNA and these histone proteins. So this set off a lot of interest, primarily Denisa Wagner's group and others, suggesting that nets can promote thrombosis in animal models, mostly small animal models in mice, and that they actually could be a mechanism for triggering contact-mediated activation of coagulation. And you can see some of the papers listed here that dealt with this. However, it depends actually on what model you look at. So while one of the ones I showed you in the last slide was using a mouse model of IVC stenosis to induce thrombosis, and in that paper they were able to show that neutrophils via nets, platelets, and factor XII all had a role in thrombosis in this particular animal model. But for example, a more recent study here from Leiden showed that in a different model where antithrombin and protein C were knocked down in these mice, they actually get spontaneous thrombosis in cerebral veins and in the liver, that while platelets played a role, there appeared to be no role of neutrophils. So depleting neutrophils had made no difference in their model in that particular form of thrombosis. And indeed, when they knocked down factor XII, if anything, it may have promoted thrombosis more. So I think one has to be a little bit careful of some of the animal models here of thrombosis, and they are, at the end of the day, only models. So the thing that interests us was there was very little of the biochemistry done in terms of how nets might actually activate factor XII. Mostly it had come from this holistic kind of animal model work, which is very important. So we set about looking at this using essentially what I'm going to show you is based on two major readouts. So we can activate the contact system however we like and see if something does activate it. We can look at 12 dependence and so on and so forth. And we can measure in plasma, particularly classic thrombin generation assay. The other approach we did was to use a purified, what I'll call a reconstituted or a synthetic contact system where we're adding the four proteins, factor XI, 12, high molecular weight kininogen, and prekallikrein in a buffer system with zinc and all the rest of it, and looking at factor XIa as a readout. So we're taking away some of the variables that you see with plasma. And what you'll see here is as a readout for measuring factor XIa, one can either detect that chromogenically with a chromogenic substrate, or we add antithrombin and heparin so that we form 11A antithrombin complexes and measure that as a readout. So you'll see some data on that going forward. But those are our two basic readouts. So it had been shown in 2007, I think, by Klaus Preissner's group that free DNA, cell-free DNA, and in our case we use it exclusively from neutrophils, will activate, they use cell lines, but this is neutrophil DNA, will activate coagulation. And just to show you initially here, in the same, as in a dose response going from 6.2 to 50 micrograms per ml, it takes a lot of DNA to activate factor 12, but it will do so in platelet-free plasma. So when you see PFP, that's platelet-free plasma, which itself does nothing. Now this is dependent on both factors 11 and 12, that if you do this in deficient plasma for 11 and 12, you see no thrombin generation. But in 7 deficient plasma, you still get thrombin generation. So this is not a tissue factor mediated event. This is a contact mediated event. So this has been shown before. This is the synthetic contact system model, if you like, where here we're reading out 11A antithrombin. Our positive control here is kaolin, as we use when we do a PTT test in the lab, or an APTT, that you see kaolin robustly causes 11A generation. And you see some dose response here with rather a lot of DNA, but it will activate and cause generation of factor 11A. So another observation that we looking at the effects of cell-free DNA was really extrapolated. I guess we stole the idea from the polyphosphate literature, that polyphosphate had been shown to amplify 11 activation by thrombin. So what we're doing here, first of all, is a plasma assay where we take very low concentration of tissue factor and get a yellow curve here showing thrombin generation. And then we are adding in DNA at various concentrations. And here we are looking at the, if you like, the extrinsic pathway triggered coagulation with potentiation of thrombin generation in the presence of DNA. And when you look at the factor deficient plasma there, you can see that the factor 12 deficient plasma, there was an enhancement in the presence of DNA, the same as normal plasma. But in 11 deficient plasma, there was no enhancement of thrombin generation, suggesting again that this was not 12 dependent, but was 11 dependent. And so again, taking this to a purified type of system, here we've got cell-free DNA. You'll see the term purified DNA in some of these slides. And what we're doing now is coming in with thrombin. Instead of initiating with tissue factor, we're initiating with thrombin, and we're making this sample rather, or the assay rather simple, adding 11 as a substrate with or without high molecular weight kininogen. This has high molecular weight kininogen. And we're looking at thrombin-mediated activation of factor 11 in the presence or absence of purified DNA, and then reading out 11A chromogenically. And so what you see here is very little 11A generation when you add thrombin to 11A. When you add the DNA without thrombin, you get very little autoactivation, I guess, of factor 11, but the presence of the two together caused significantly more, at least, 11A generation. So this is compatible with the last data showing that tissue factor in plasma or thrombin in a buffer system, that this is being amplified by DNA. So the other major component of the chromatin, of course, is histones, is the histone proteins. So it's also been shown that histone H3 and H4, this is primarily from Chuck Esmond's group, showing that these trigger thrombin generation, but this is platelet dependent. And again, we're reproducing that here, so I'm just going to walk you through this. If you look at recombinant human H3 in platelet-free plasma, here's our positive control with kaolin. Nothing happens. This is micrograms per ml. And recombinant H4 in platelet-free plasma, nothing happens. But in the presence of platelets in platelet-rich plasma here, you can see that both H3 and H4 amplify the thrombin generation. And this was shown by Chuck's group and suggested that this may be dependent on TLR receptors on platelets. And this is true for both H3 and H4 here. Now, we have access through actually a spin-off company in the biochemistry department at UNC that is interested in epigenetics to some of these reagents, which has been very useful. And so what we're looking at here is the octamers. This is recombinant octamers. And you know that in nucleosomes that these homodimers of H2B, H2A, H3, and H4 assemble into an octamer, essentially as the core protein component of a nucleosome. So unlike the fact with recombinant single H3 and H4, which generated thrombin, when it was assembled in an octamer like this, in a more complex structure, we did not see any increase in thrombin generation in platelet-rich plasma. So again, the data are consistent with what's been shown in the literature. Just to summarize that, in platelet-free plasma, DNA will activate the contact system, and that histones, at least single histones, will activate thrombin generation in platelet-rich plasma, but not when they're assembled into an octamer. Okay, so we then wanted to look at the effects of nets in our system. So this is just to show you that this is using a fairly classical so-called suicidal netosis, where you're using PMA or ionophore for longer periods. And you can see here that there's this extrusion of chromatin, which disappears with the addition of DNAs here. And also, one of the hallmarks of NETs formation is the citrullination of histone H3 through the PAD4 enzyme in the neutrophil, that these arginines are converted to citrulline, and this is an antibody looking at citrullinated H3 histones here in the extracellular space. So this is basically essentially fairly standard readouts for NET formation in 2016. So we took, for these experiments, and we did this two different ways. We isolated neutrophils, and I will say that if you're going to do these experiments, you have to be absolutely fastidious about making sure there's not platelet contamination, because several groups have shown that platelets can contribute to NET formation, and platelets can do things certainly independently of neutrophils and confound the experiments. So these are very pure neutrophils that are isolated from blood. And again, what we're looking at here is either where we make the nets and then leave the neutrophils in there. We're calling this in situ nets or intact nets, or we wash the cell, the nets free of cells as best we can. I mean, these are kind of, as much as we can, we make it cell-free and retain the nets. So I'll call these washed nets. So what you're looking at here is that the intact nets with the neutrophils in there did not trigger thrombin generation in platelet-free plasma. And so what you see here again is the positive control, the kaolin. The blue, which you can't see, is along the line where we added neutrophils. And then the other three lines, which are also flat, are when the neutrophils were induced to produce nets in the presence, and then plasma was added. There's no thrombin generation here at all. And then because we also wanted to look at so-called vital netosis, where we didn't necessarily use PMA or ionophore, but used LPS, essentially very much the same thing, except that what we're looking at here is platelet-rich plasma here, that you essentially don't get anything in platelet-rich plasma too. So we're essentially covering, if you like, the PFP as well as the PRP situations. The same was true of when we looked at the wash nets. So here we have tried to wash away the neutrophils, but again, when the nets were added to plasma, nothing occurred. Now, what you're looking at here is this is where we then took the wash nets and then made DNA from it in this red one. And the DNA is still active when it's made from the wash nets. But when it's in the presence of the whole wash nets here, it's flat. So we're seeing a dichotomy of the results here between a different result with DNA when it's free or histones when it's free, and then the intact nets. And this is essentially also using the biochemical system, if you like, the synthetic contact system, where we have the 4 proteins here. We have kaolin as our positive control. Where we're using unstimulated neutrophils, nothing occurs. When we use PMA or A23187, this is a somewhat cleaner system. There's no factor 11A generation in the presence of those nets. I think this is important. It really gets at whether a neutrophil can activate coagulation. I look at it that if we know a monocyte can, if we activate a monocyte, we can make tissue factor, we can measure tissue factor biochemically, and we should be able to do that with a neutrophil, but at least in our hands we're having trouble doing that. So the other situation I showed you is that cell-free DNA will amplify tissue factor, low concentration tissue factor initiated thrombin generation. And here we're taking out factor 12A by adding corn-trypsin inhibitor. But here we really don't see much in the way of, here's tissue factor alone in the green, and the other 3 are where we're either using unstimulated neutrophils in there or neutrophils that have been activated with PMA or A23187 to make nets. And here we're measuring this actually, there's no enhancement of thrombin generation here. So what is going on here? Well, free DNA has this very negatively charged phosphate backbone, as you know. But when chromatin is formed or nucleosomes are formed, we see the octamers here that I mentioned already of the histones, and DNA is wound around this. And of course, this is very much associated with a mutual neutralization of charge. And so we think this is probably essentially just a charge issue. So in order to really look at this further, what we did was to take isolated neutrophils, and then we purified DNA from them and purified the histones. And these are kits, essentially a QIAGEN kit and an Abcam kit to purify the histones. And then we recombined them through the salt gradient dialysis here to allow chromatin reconstitution. So we took them apart and then put them together again, and then compared the effect of 3 things: the purified DNA, the reconstituted chromatin, and the purified histones here on our assays that I mentioned to you. First of all, we wanted to make sure we reconstituted nucleosomes, and there's a commercial Roche kit that will do that, and I'll show you those data in a minute. Then we wanted to make sure that the DNA was there or not. I'll show you that. And then the 2 readouts I mentioned earlier, the thrombin generation and the chromogenic synthetic contact assay. So first of all, to confirm that we had been able to reconstitute the chromatin, what you get here is a positive and negative control with the Roche kit. It's an antibody that detects it's an ELISA that with one antibody detects DNA and the other is a polyclonal against histones. So essentially, in order to be detected, it needs to have both components in there. So the purified DNA from the neutrophils, we got no signal, purified histones, but then when we reconstitute it into chromatin, we see the nucleosomes here. And when we looked at the effect in platelet-free plasma, what you see here is our positive control kaolin, our negative control, which is the platelet-free plasma. And the DNA before or after dialysis led to the same thrombin generation. Histones in platelet-free plasma did nothing, and the chromatin did nothing here in terms of thrombin generation. And then also, since we had these, we looked at the buffer system, and you can see that kaolin is here, our positive control, negative control. The purified DNA, as before, was bioactive here, or procoagulant, if you like. The histone, neither the histones nor the chromatin were active in terms of platelet-free plasma. And then finally, the assay also looking at the effect on of potentiating 11 activation by thrombin. We used our substrate, which was factor 11, our enzyme, which is thrombin, and our readout, which is 11A generation. You can see that the purified DNA once again did this. The histones really did not, the chromatin did not. So again, our various components were intact when they were purified, but when they were reconstituted into chromatin, we didn't have an effect. So to summarize this part, the purified DNA triggers thrombin generation in plasma, and that's platelet-poor plasma, platelet-free plasma, but it'll also work in platelet-rich, independently of the presence of platelets, in other words. And when we look at histone proteins, we can see thrombin generation that's platelet-dependent. When we recombine these into octamers and change essentially how the histones are presented, we fail to see that they activate coagulation through platelets. And when we reconstitute DNA and histones into nucleosomes, we also see that the procoagulant activity is lost. And so also when we look at the most complex structure, which is nets, and there's a lot of other stuff too, including all the proteolytic enzymes, the elastase and so on, and cathepsin G and what have you from neutrophils, we also didn't see any direct activation of coagulation in vitro. Okay. So I'm going to switch gears with about 4 or 5 slides with a slightly different angle on, because now that we've gotten into the interest in what it is that may be activating factor 12 in vivo, let me give you a couple of other potential ideas. So I'm going to talk a little bit about microparticles here. And without having a lot of time to go into background, I think most of you are quite aware at this point that microparticles are shed by all cells when they're activated or undergo apoptosis. So this is an in vivo model. This is patients. These are students from the University of Vienna who are infused with endotoxin. Now, it's not compulsory apparently to do this, but they do get rewarded with this, for this. So this is a human endotoxemia model. And what we're looking at here is the total number of microparticles goes up. This is not differentiating the source and then comes down again. When you look at the source, you've got red cell microparticles, platelets, monocytes, and endothelial cells. And this is done by flow cytometry. You can see actually that the increase is really in red cell and platelet microparticles, which was somewhat surprising to us. And this was the interesting data from our point of view, that if we then spin out those microparticles from the plasma and add them back to various types of plasma, that's what you're looking at here, and we record the clotting time. So if we take normal plasma, spin out what's circulating in terms of microparticles, put it back in that plasma, you get clotting times up here. The paired samples on the squares are the 0 time baseline, and the round circles are at 3 hours after administration of endotoxin. So what you see here is that there's a shortening of the clotting time when the microparticles are added to normal plasma after 3 hours of endotoxin. But this doesn't say anything about the pathway. It doesn't really shift in factor 7 deficient plasma, but it did in factor 11 deficient plasma. So if you look at, say, the baseline at time 0, and time 0, you can see that actually this was really affected by the factor 11 here. So whereas the 7 really wasn't affecting it. So just to cut to the chase, there's plenty of data that in the endotoxemia model that monocytes are getting activated, the tissue factor is being produced, that you can inhibit this with 7AI and tissue factor antibodies in baboons, and that we think this is primarily a cell phenomenon, that the majority of tissue factor will be circulating on cells, but the microparticles may be produced, and we can measure these with tissue factor on them. But the real effect of the microparticle fraction in this particular model seems to be more driven by a non-tissue factor dependent mechanism. So again, it doesn't say anything about the relative contributions of tissue factor and non-tissue factor dependent mechanisms, but certainly the cell bound seems to be tissue factor dependent. This is probably not. And so this is my last slide, just to show you that what we're doing now is actually looking at a much more purified system. Here we've taken red cells and made sure they're pure and make microparticles from them. And in all cases, the blue is the baseline plasma we're using. Microparticle-free plasma does nothing on its own. When you add red cell microparticles, we get an increase in thrombin generation here, which is obliterated by the addition of corn trypsin inhibitor. So again, in 12 deficient plasma, nothing happens. The red cell microparticles do nothing in 12 deficient plasma, but when we add back factor 12, we get thrombin generation. The same with factor 11 here, that in 11 deficient plasma, nothing. When we add back factor 11, we get thrombin generation. And over here, in 7 deficient plasma, there's nothing, and the red cell microparticles do their thing in 7 deficient plasma, suggesting that this is not 7 dependent. So this is really essentially where we're looking at now, is looking at some other, particularly red cells we're kind of interested in terms of their effect on contact activation, and there are candidates for, and we can discuss that in the discussion if you'd like, for red cell or membrane or microparticle components that may activate the contact system. So I'll stop there and thank all the people who have helped with all of these experiments. I forgot Mac Monroe's name on here. I'd like to thank him as well. These are people in my lab, Raphael's lab, and Bernd Gilmer for his group in Vienna. Thank you very much.
Jacob Rand from Cornell, New York, USA. And I'm seriously thinking about moving here. Nigel, so what you've demonstrated in your dissection of the nets and coagulation is very intriguing and novel. But how do you view it in the context of biology? Are you discounting any connection with coagulation, or do you have some other view on this relationship?
I happen to have a backup slide here that
this was not planted.
No, but this is just a cartoon, right? I just pulled this out from a review, and I think, first of all, there's two things, Jake. One is we talked about assembling into complex structures nucleosomes and nets, and this inhibits 12A generation. What we haven't done, and what nobody knows, is what happens as you disassemble nets. Could it be that you release enough cell-free DNA to do it? You know, so it could be the other side of the coin. The other thing is that, and there are data on this, and there's some, actually the cardiologists are hot on this in the coronary arteries, that actually the nets may be important for localizing tissue factor and maybe even microparticles, which we've talked about for a long time, and that this tissue factor may not necessarily be coming from the neutrophil or having anything to do with the net structure, but it's just a sticky glue where they localize. So no, I don't think nets are excluded as being important, but I think that mechanistically when you're designing, therapies, it's important to realize what you're targeting, which could still be we don't know what net breakdown looks like, what the length of the DNA is. Is it enough to trigger 12 or is it not, and so on and so forth. So I'm not sure. Two questions here.
Jake is a cadet from UBC, CBR. So very interesting talk on nets, and I congratulate you on that. And the question is actually like, there's a lot of literature on the nets activating platelets, and in your essays actually you haven't talked about the role of polyphosphate, which is released from platelets, might be, which is together is activating the coagulation there actually. So could you comment on that?
Yeah, I mean, this is again where you start getting into the complexity of the system. Then I think of it, conceptually as two different things that platelets may contribute. One is that they themselves, through polyphosphate, independent of neutrophils or nets, may be able to activate the contact system. And I think Jim Morrissey's group and others have, shown that that's certainly a possibility. The other, and Rene and others. The other thing is that nets, that neutrophils, that platelets very likely have some role in promoting net formation, and then however that becomes procoagulant in the neutrophil. There's evidence that the various, DAMPs, HMGB1, for example, was produced by platelets, may activate neutrophils or be cooperative in the formation of nets. But then it begs the question, how then does that become procoagulant? So I think platelets may be able to do this independently or may cooperate with neutrophils to produce more nets. I think it gets really pretty complex. So.
Thank you. Wearing your sort of quasi-clinical hat, Ross had a PhD student working on factor 12, and when the 11 bypass came in, it was dropped. But Ross pointed out that factor 12 is conserved, so it must have a major biological role.
Yeah.
Two things were kicking around at the time, not that we studied them. One was that it was a low-grade regulator of fibrinolysis.
Yep.
And you didn't actually address it. I don't know if you want to address it briefly. The other thing, probably sort of no clinical consequences, alleged that people who were 12 deficient were prothrombotic. Could you comment on either of those?
Well, I happen to have another backup slide here. So yeah, 12 does a lot of things, and this is the really fascinating thing about the molecule. Is it the alpha form, the beta form, or some other mechanism that regulates what 12 decides to do? I mean, we've only talked about this top left pathway at this point. As you say, there may certainly be a role in fibrinolysis. There clearly is a role in the kallikrein pathway. Again, that I know of, the company that made the antisense oligonucleotide for factor 11 is now making one for factor 12. They're not targeting thrombosis, they're targeting hereditary angioedema. So this is really clearly important. And then I would have to defer to Ed and others about the complement system and the role of factor 12. So it's really a fascinating molecule in this respect. That was the second part of your question was? Yeah, actually, it's been bandied around for quite a while, I would say, whether 12 deficiency is associated with thrombosis. Yeah. So a couple of years ago, I did an education session for ASH and reviewed all the literature on this, and I I think, and this is published, but my conclusion was that really from the real prospective cohort studies that you can't really say. The most interesting data are from Vienna, where there's this U-shaped curve actually that you know, that it may be biphasic, it may depend on the level of factor 12. So, but at this point, I'm not sure that there's enough evidence to say that 12 deficiency is associated with thrombosis. So.
Don Brooks from the Center for Blood Research. We've always seen pictures like yours with artificial surfaces with negative charges activating factor 12. If that solid artificial surface was positive, does it do that? Do you know?
Does an artificial surface do that? Or?
No, with a positive net charge.
I don't think so. You would have to ask Earle that. We haven't looked, but I don't think so. I mean, I might be wrong, but I don't know of anything other than, you know, negative charge that really causes autoactivation, and so.
Has it actually been done? And Earle, do you know?
No, I don't. I haven't seen anything on that.
Okay. That's what I thought.
Yeah.
So the interaction between RBC microparticles and factor 12, do you think that plays a role in bleeding, or would it only be during sepsis or some other process?
Yeah, I don't know. I mean, you know, this has been kicking around. Is this a good thing or a bad thing? And does it set the tone? Are the microparticles actually, you know, responsible for some background turnover of factor 12, which is occurring? I don't know whether it's, you know, whether they're detrimental or helpful. We always go back to Scott's syndrome, where these patients don't make microparticles that have a mild bleeding tendency, but whether you can really say that's mediated through this particular interaction or prothrombinase effect or something else of those microparticles, I don't know.
Nigel, behind you. Ed Prydesdale. I didn't quite understand your experiment where you compared the in situ versus the washed nets. Yeah. I didn't understand the difference there.
Well, what we were trying to do, Ed, was to, and it's very difficult to do actually, was to separate the neutrophil from the net after the net had been made by the neutrophil. In other words, spin out the cells and hope that we got the nets without the rest of the neutrophil. But there's so much junk in the nets already that we actually got the same result regardless, but we were trying to make it cell-free.
Oh, I thought that you got a different result.
No.
The wash versus in situ.
No.
So that was my misunderstanding.
Yeah, no.
Okay, thank you.
Any other questions? Okay, well, thanks very much, Doctor. So it's my pleasure always to introduce Shannon Jackson. Dr. Jackson is a hematologist at St. Paul's Hospital, and she's the director of the BC Provincial Hemophilia Program there, where she leads a tremendous group, very unique in Canada actually. It's so wonderfully cohesive and interactive with patients and with colleagues, professional colleagues. Shannon trained in her medical training at the University of Calgary, and then went on to do hematology, and then received a fellowship to do her hemophilia or hemostasis fellowship there under the tutelage, I guess, of Manchu Poon, who's well known in the hemophilia world. She was recruited happily to Vancouver about 7 years ago, where she took over a program, and she's really expanded it tremendously. Her title is personalized treatment. Actually, Shannon from my perspective, she really introduced personalized medicine to the hemophilia world before personalized medicine was a sexy topic. She's really a leader in the field, and she runs a terrific program. I'm pleased for her to be here.
Thank you so much, Ed, and it's a pleasure to be here at Earle Davie again, up at the podium, and I'm thrilled to come with such a great round of speakers all day. So personalized treatment approaches for hemophilia. Just want to say most of you know hemophilia, that it is an inherited bleeding disorder associated with a mild, moderate, or severe deficiency of the most common being factor VIII or factor IX, hemophilia A and B respectively. In Canada, we're very privileged to have the availability of treatment, and we use recombinant factor concentrates, either factor VIII or factor IX, supplied by our health system for patients who don't have inhibitors. And about 90% of this factor is infused in the home, so patients are self-infusing it, specifically those who have severe or moderate disease. And that's largely to prevent some of the complications you see on the slide, which are historical, fortunately, and still do exist to some extent. So bleeding into the joints, resulting in arthropathy and damage and ultimately disability, and worst case scenario, bleeding into the brain. We still do see some younger individuals who develop arthropathy, and this is where personalization comes in and trying to prevent those complications. So I was fortunate in 2012, I guess about 3 years after I came here, to present a topic that really looked at our evaluation of using prophylaxis or preventative treatment in the adult world. And this had previously been primarily a pediatric practice called primary prophylaxis. And I shared with you back then a little bit about our experiences in adjusting this primary prophylactic approach in children to the adult world. And when I did that, I introduced you to a few patients, virtual patients this time, and one of them was Mr. Earle, and he was a gentleman in his late 30s who hadn't had much access to treatment, and so had developed some arthropathy shown by the little red dots there. So both of his ankles, his right knee, and he had some problems in his hip. And he'd been having about 4 or 5 bleeds per month before we put him on standard prophylaxis. And then I shared with you a story of how we had adjusted his prophylaxis, and he actually went on everyday prophylaxis, and he'd had no bleeds for about 2 years when I shared with you this in 2012. So that was personalization then. And we had learned through our patients here in BC that adults could definitely use prophylaxis to prevent bleeds, sometimes entirely. They could adjust it and personalize it and adapt it as needed. And we were really beginning to see that there was an improvement in overall well-being, and some of our patients were able to exercise more in spite of having fairly severe joints at baseline like Mr. Earle did. So today I wanted to update you and provide you with a little bit more opinion-based experience and how I see personalization now in our clinic and in the future. And I'm going to discuss a couple of different patient-reported outcome measure approaches, again a little bit more opinion-based this time, and then contemplate with you a future. And I know throughout the day we'll probably hear about exciting developments that will be pertinent to hemophilia, but how those are going to be important for personalization in our clinics. What I'm not going to talk about is something entitled precision medicine. This crowd will love precision medicine. So you find a molecular or genetic or protein defect in a specific patient and you target a treatment specifically for that patient. And an example is a BC woman who was reported in the Vancouver Sun and subsequently in the medical literature who was given a blood pressure drug called irbesartan, an angiotensin receptor blocker. And this put her multiply refractory colon cancer into remission within 5 weeks. And a publication, she remains in remission at 10 months. And this stuff is extremely exciting, and I think a lot of our excitement in hemophilia will look forward to genome editing and that type of thing that can be extremely personalized, but that's not what I'm going to talk about today. When you're thinking about personalized medicine versus precision medicine of course takes into account patients' goals and preferences, but it's not the primary mandate for the treatment decision. Personalized medicine, on the other hand, is really taking into account what the patient's goals and preferences are, and specifically the impact of a treatment choice on the day-to-day living for that patient. It takes into account the personal aspirations, the clinical course and experience of prior treatments in that chronic illness, and lifestyle preferences that may change between seasons or years. Now we look out to the physicians in the audience, and like me, when they first started to read this literature, they're saying, well, this is important, but I always do this. We always have conversations with our patients about their preferences, what's going to work in terms of treatment outcomes and discussions. So I'm not saying anything new here, but what I'd like to do is challenge us to think about this a little bit differently in terms of how we measure how much impact our decisions are having on our patients and how we're receiving information back from them about this. So there's been some groups of hemophilia treaters who've gathered and looking at what are the right outcomes to collect in hemophilia from the perspective of clinical trials primarily. And there was a good quote in one of the articles that was authored by Kathleen Fisher, and I'll read it to you. While both patients and clinicians are interested in achieving optimal outcomes, it is the patient that faces the burden of treatment, and this may lead to different perspectives. And I fully agree, it may lead to different perspectives on what the most valuable outcomes are. And there's a growing interest in looking at what we call value-based healthcare. And Michael Porter, through Harvard University, does some nice articles related to this. And it really looks at not healthcare as a volume-based service or as a service to follow guidelines and look at process, but really we should be looking at defining value. And the value should be defined as the patient outcomes achieved versus the relative cost to the system. And I know that's very theoretical, but I think there's some important underpinnings here. And part of that is actually evaluating the outcomes you can achieve. But before you can do that, you have to define the outcomes, you have to measure them, and then you have to assess them for their value. And I think this is where we need to go with personalization, especially in hemophilia. And that's where it becomes important to take the patient-reported outcome component of care in collaboration with the standard disease-specific outcomes that we always measure. And I think hemophilia is a really unique situation because we know our patients with great support of our clinics across Canada and internationally where care is available. They're really responsible for detecting the bleeding episodes at home or at work. They are responsible for determining if they're going to seek an infusion or if so, what the infusion schedule is going to look like, and really are the ones making the decisions about return to normal activity or not after a bleed or a surgery. And we've worked hard, I think, in our hemophilia clinics to support that autonomy because it's very important for trying to seek a normal life. However, I think what that puts us as clinicians into the position of is it's harder for us sometimes to appreciate the burden of that treatment on a day-to-day basis. And we really don't have a good way of measuring that. And here's just a synopsis of some of the common clinical things that we collect in our hemophilia clinic. They're also the premise of most clinical trials. So looking at things like joint outcomes in various ways, we can look at that, looking at annual bleeding rate, establishing inhibitor status and risk, and as well, unfortunately, still looking at treating and monitoring co-infection. And in general in clinic, and it's attempted in clinical trials, we try to measure some form of adherence to what the treatment is and how closely it's been followed, as well as assess quality of life. And here are some areas where we can use patient-reported outcome measures. And when I spoke in 2012, I was really trying to highlight to you how we were using some of our tools and in a personalized way to adjust our outcomes, such as bleeding outcomes, such as joint health and that type of thing. So we're quite good at this. We can personalize in this way when we're looking at the standard clinical outcomes quite well. So I wanted to provide now, before I go on, to talk a little bit about how Mr. Earle is doing now. And he's still with us in clinic. He's got severe hemophilia A. Through his prophylaxis that he is still on, he's still having virtually no bleeds per year. Joint status is stable. His co-infections and inhibitor status are unchanged. But does that really tell us what we need to know about how he's doing? So let me just reread to you a slide that I created then about his testimonial. And he said, since I started prophylaxis treatment, my life has changed. I get more confidence when I go to work in the morning and I don't have to worry about bleeding in the afternoon. I can focus on job and family. And I think that's great, but what I don't have for you today is a way of reflecting to you that in an outcome measure. All I can do is reread you the testimonial that he said a few years ago. What we want to do is look at these personalized outcomes that give us a better sense of the value of the treatment that he is implementing and the value of treatment that as a health system we are supporting. And that includes things like participation in social, family, societal wishes and obligations, physical activity, and things like work productivity as examples only. So I think it's time for us to take this patient perspective formally into our clinics and make sure that we're accounting for those things that are harder for us to do when we only see our patients, 3 times a year, twice a year, even once a year sometimes. And this is just an example, but these may be spheres of looking at their goals with respect to school or work, with their goals with respect to economic goals, physical activity, lifestyle, and then the participation that I already mentioned to you. So I see a future where we need to have some more formalized crosstalk between these two systems. They've always been there, but we're not measuring them very well. And we've got more and more tools at our disposal in which we can do this. I've listed some of them along the bottom. They're related to standard clinical outcomes like bleeds and joints. We can look at different factor replacement. We can alter the half-life, the infusion timing. We can look at pharmacokinetic altered infusion strategies. We have all of this at our disposal. And as I'll show, we have some future alternatives that may make this look a lot different. But I think we need to balance that by having some better ways to support our patients and to look at their outcomes when it comes to things like autonomy support, vocational adjustments and counseling, psychological support. These things just really aren't funded in terms of most hemophilia clinics in Canada, nor do we look at these outcomes to try and generate the funding we need for these. And so patient-reported outcome measures are one way of personalizing care beyond what we're doing now, and they ensure that our care remains very patient-centric as it is already. Interestingly, the FDA, the European medicine agencies, and Health Canada are a little ahead of us in that regard because for almost the last 5 to 10 years, collecting a patient-reported outcome has been a mandatory requirement for new pharmaceutical agents studied in clinical trials. So it's interesting, I find, that those are part of the clinical trials. We collect quality of life data, we collect a number of questionnaires, but we really haven't translated that into clinic in terms of when the products come to the patient level. What are we measuring and how are we showing improvement? And if we were going to do that, there's really no guidelines on which tool to use, and perhaps we don't have a level yet of personalization that we need. So I'd like to switch now and talk a little bit more about a practical application of patient-reported outcome measures. So many of you in the audience will have heard of a lot of patient-reported outcome measures that are standard tools. One example is a quality of life questionnaire. There can be other questionnaires related to work and physical activity and pain and that type of thing. A common health-related quality of life questionnaire is the SF36, a very generic tool, can be used to compare individuals to large populations. We also have similar quality of life tools applicable to hemophilia. And these are great, but they're not necessarily what I would think in our clinic are going to be the most useful tools to get patient-reported outcomes. They're not really adaptable to an individual patient. They're fairly time-consuming, so I can't imagine adding on 4 or 5 questionnaires to each clinic visit. And what we end up getting often is an overall score that reflects an additive phenomenon of a number of different domains that may look at physical function, emotional functioning, role functioning, that type of thing. And the overall score may not detect subtle changes in very important categories for that patient in order for us to assess how things are going. And so even though they're mandated in clinical trials, I don't necessarily think these are the right tool for personalization in the hemophilia clinic. One type of personalization that we've started to look at a little bit in Vancouver, and I wanted to share with you for that reason, is something that we all giggle when we say the name. It's called GAS, Goal Attainment Scaling. It's actually not so much a questionnaire, but it's a sort of a process developed in the 1960s, a collaborative process between a clinician generally and a patient together. And it's really designed to set goals such that you can measure the extent that the treatment results in attaining that goal. And it has to be that the goal relates to somehow the clinical or functional aspects of that condition. And what this method can do is detect very small, but importantly, very relevant changes for that particular patient that may not be relevant for other patients, even in a similar category, and that may change over time depending on how things are going. Obviously, a limitation of a process like this is it needs a little bit of training and work, and it's operator dependent in the sense of working with it. But I'd like to share with you an example just for fun, so you can see how this may work. It also requires fairly short-term follow-up as a necessity to do the scoring a little bit later down the road and compare pre and post. Usually, 3 or 4 goals are selected at a given time, or it could just be one goal. It's not mandated. So a selected number of goals are selected related to a topic germane to that particular patient at that time. It's been largely studied in dementia and diabetes and some chronic rehabilitation settings, but I'd like to share with you an example of dementia because it's something that I think we'll all be familiar with a little bit, and I certainly forget a lot, so I can relate. What is done here is there's sort of 5 scales, so you go from -2 to +2, and what you do at the visit is select baseline. And baseline is something that should be improved upon. We would like to see a positive change in a baseline. It could be just about anything. In this case, I'll present to you memory loss. So individuals with dementia often ask repetitive questions. So they ask a question, when are we going for dinner tonight? They get the answer, and then they ask it again and again and again through the day. So if we use that example as someone who forgets and has to re-ask a question like that once an hour, that would be their baseline, -1. And then together with the patient or the caregiver or the family, you establish, well, what would 0 look like? It might look like forget 6 times a day instead of once an hour. And that's a good thing, that's a goal, and that might be enough of a measurable change for that specific treatment that may be used for the dementia to make a positive difference and to add value to care. And then generally you score a little bit higher and a little bit lower with outcomes that might be achievable but maybe perhaps a little bit of a dream. But you still do it. So it might be 3 times a day for somewhat better than expected, the green arrow. And then the best possible outcome, still technically achievable but probably fairly unlikely, would be the score value of much better than expected or +2. And that patient might forget once a day, sort of like me. But the negative score is also important because it's important to track slipping too. So this is done and then evaluated in a given time depending upon that goal. So there's been some work in hemophilia using this gas technique, primarily from the basis of supporting hemophilia treaters to learn the technique and to provide some resources for us. So a group of patients and families and healthcare professionals as well as gas experts developed an online platform and a little bit of a guideline for clinicians. And I'm proud to say that our clinic has been participating in an ongoing North America study to look at this in the clinic, primarily heavy lifted by Deb and Sandra Squire, who are here today, who've really worked with the patients and I think very much enjoyed this process with some experience that Sandra had already had doing this. So maybe we'll be looking at that in the future as a way we can evaluate some of these new treatments that are coming down the pipe. So in terms of treatments, what will personalization look like in the immediate future for hemophilia A? Well, in my opinion, we'll still continue to have options such as extended half-life factor 8. It may get more extended with abilities to modulate von Willebrand factor. And we will be looking at likely alternatives to factor replacement for prophylaxis. So patients like Mr. Earle will have different options. And these options to date in early clinical trials do look like there may be acceptable bleeding rates on prophylaxis with a route of administration that may alter from the current traditional intravenous infusion and become more like insulin or subcutaneously infused. So this may be possible. And here I list just a few candidates that are in clinical trials right now that might be in our grasp in the next few years and will certainly force us to have a discussion about personalized outcomes and what we would desire for each specific patient. For hemophilia B, much the same. We will have extended half-life products. There's more coming to the clinical arena. We also will have alternatives such as gene therapy, which will be realistic, I think, in the next few years in Canada, as well as some of those therapeutics that I've already mentioned to you already. So as clinicians and patients, when we're faced with personalization decisions, how do we choose which treatment? And I think really it's going to come back to the personalized care and looking at which agent would provide the best option or opportunity to achieve a standard outcome, which agent will, for the patient, be able to give the most value and benefit as we should and will likely have to measure as a patient-reported outcome. And in BC, I'm happy to say I think we're going to be starting to formalize collecting both standard outcomes as we've done and looking at some innovations with collecting patient-reported outcomes next year. And I think that's going to best reflect, hopefully, what I'll be able to talk about in a few years if you're asking me back to talk about how we've been able to look and reflect that type of care. So in conclusion, I just wanted to provide some thank yous to some folks here, specifically our patients, all of them who have worked with us over the last few years to individualize and personalize prophylaxis, and they continue to teach us things every day in clinic. To both Vancouver hemophilia teams who continues a willingness to learn and to innovate and to try new things, and also to Ed Conway and Hannah Kim for continuing to organize this wonderful event on its 10th anniversary. So thank you. Absolutely. I couldn't agree more. I feel very deficient in clinic when I'm faced with, pain of many different etiologies. And as physicians, we're basically trained well in the pharmacologic aspects, but we're not necessarily holistically trained in how to manage it. But moreover, that's once you've really defined it. And I think the ability to measure pain is a problem. Most of the clinical trials use a visual analog scale or some kind of very short-term lens on the pain that's going on. And we recognize that over the time and over the seasons and over the day, the pain can fluctuate, and we're not really very good at assessing that fluctuation. And I think that for our clinic going forward, I would certainly like to see us have a very easy way of reflecting pain on a more ongoing basis. So it's not like a one-time tool that you just ask, but you actually measure it more continuously. And then we can look at the ebbs and flows and flux and try to come up with holistic treatments that are partly pharmacological, partly probably biofeedback, partly strengthening and working with optimizing muscle function, and be able to kind of turn that around and say, well, there's an outcome that we could measure and show over time that we've decreased the amplitudes or the sort of median level of pain. I think that's a much better way than just a one-time tool.
So Shannon, just further to that question, can you just comment on the costs of your prophylaxis regimens versus your non-prophylactic approaches, and because that has bearing not less here because we're not under the constraints maybe of a third-party insurer as Nigel refers to. Yeah, I mean, I think the payers certainly in Canada are more provinces, and they're certainly very interested in the cost. And I actually didn't say this, but in 2012, when we had looked at the cost of Mr. Earle's personalization, we actually decreased the factor utilization by about 40% from historic by using prophylaxis, by using it in a very tailored way. And I think that the payers, they like to hear that, and I think that without good care, I think, automatically adds value. If patients are more functional, if patients can work and contribute to the societal structure and pay taxes and all those things, I think it's a win-win. And so I think it really behooves us to sort of prove that the value of those products goes beyond a bleed reduction, because I can have lots of people having 0 bleeds, but they don't do anything. They're not going to get up and go to work, and that's not at all what we want. We want to prove that the investment is causing 0 bleeds and actually contributing to patients who are able to participate in society. And I think that's where the patient-reported outcome measures could, if properly devised and used and personalized, could actually show that. But there's different people who are interested in patient-reported outcome measures. So we have the government, the payers, we have the scientists, the clinical trials, and we have the patient at the end of the day and their family and how that works. So to address all of those in one go is going to be a challenge.
Shannon, over here in the corner where I belong. I know that you're switching over to long-acting factor. Are you starting to get feedback on quality of life from these patients?
We're certainly collecting some information, but it's not yet, in terms of patient-reported outcomes, it's not yet looked at in terms of analysis. It's pretty early on, and I think most of my feedback has come through clinical trials and participating in those, and just again perceiving that there is a holistic kind of benefit. But one of the difficulties is that we have in our quality of life tools that we have, there's not necessarily a accepted change that is meaningful to the study or the payer. So patients may be feeling better, but we're not necessarily able to well measure that in our existing tools. And I think again that's going to be related to which tools we choose and how we choose to analyze them. But yeah, we're seeing an improvement. And I think when we're talking about switching from intravenous replacement to subcutaneous, there's going to be a whole different type of improvement that we'll see. But the question is going to be, how do we convince our payers that that's going to be a worthwhile investment when already we're achieving standard outcomes that are pretty good. So I think we're learning what we will with the extended products that we'll have to translate when we see some of these factor replacement products come to the clinic.
Over here. Shannon, that was really interesting. And I'm wondering, do you ever have conflicts, as you do personalize more and more with what you see as a treater as appropriate, goals or based on cost? An extreme example might be, let's say you had a patient who said, I want to play high school football and I want to be, get factor so I can go out there and hit, because generally that's something that we strongly discourage. And it's an extreme example, but I don't know if there's others that you might have run up against.
Yeah, well, I mean, yesterday I had a young man who fractured his radius playing rugby, who has hemophilia. So I don't think it's happening. Patients are doing these things, and as you'll hear soon, many patients have already been doing these types of things. And I think my feeling is that we need to support as much as we can in terms of activities that are reasonable. And then there's sort of a few that are a little bit, off the charts, like rugby and, crash football and that type of thing. And I think, as much as we try to discourage that type of risk taking, I think we're all very much engaged in trying to optimize the factor levels if that's going to be activity happening anyway. And John will have more experience with younger boys doing that, but it is sort of a fact of life, and it does provide, a quality of life and something that at least in the short term fulfills the patient's personal goals. It may not work out in 20 years when we have bad knees and ankles as a result, but that's something that we have to talk over. Thank you. Thank you.
I'm just going to delegate. Our next speaker will be introduced by Shannon Jackson. Could we turn this mic up a little bit? And when you do answer questions or ask questions, please use that mic and hold it close to your mouth so that we who have less good hearing, is that English? Can hear. All right, Shannon.
Thank you, Ed. I just want to commend you over the years for having the vision to invite patients to come and share with us at the session. I think all of us remember the stories and the experiences that are shared so heart-feelingly through people like Tim, who are going to join us today and talk about their experiences. So I met Tim a number of years ago and certainly consider him one of the forerunners in allowing us to adopt prophylaxis and hear about his goals and experiences. And it's just such a rewarding experience to have him now be able to share that with you. And I don't want to take any of your thunder, but he's recently retired from teaching, a long-term prestigious career in teaching, and he's certainly taught us a lot in the clinic, I can say from my heart. So thank you, Tim, for coming up to share with us a little bit about your experiences. I think you can go, oh, there he goes.
Thanks, Ms. Shannon. Just here? Yeah, sure. We'll put that picture up there. Good. Good. You might have made a mistake turning up the microphone for me because sometimes I have a rather loud voice. So I'm under a lot of pressure today because 10 minutes. I see we're a bit ahead of schedule. Ed, I can put you back on track really fast. But I'm happy to be here. Pat, now first, let me prove my authenticity as a hemophiliac. There are the flexion deformities in the right elbow and the left elbow. They appear straight to me, but if they're not straight to you, the right knee, do you want to see it bend again? Yes, Sandra Squire, our physiotherapist over there, she says, Tim, it's now down to 0. But then I convinced her, no, she got up to 3 degrees of motion. There's no, for a long, long time, my left ankle has had no motion in it. And my right ankle, my current pain in life, is catching up rapidly to my left. But my shoulders are great. My hips are good and rather attractive. So things are good. So how did I get this way? So it really is an honor to speak today because at my table is Dr. Davie, and my life somewhat parallels his research because he received his PhD in 1954 from the University of Washington, and I was born in 1954. And by the time he and Dr. Ratnoff had published their groundbreaking work, that was 1964. I saw earlier on a slide that was September. But this is probably July of 1964. And this is my hemophiliac family. So you'll see me, I'm that cute little guy on crutches. And then beside me is my sister Judy, a carrier. My beautiful mother Betty, she's obviously a carrier. Cousin in the sling, and we were often slinged in those days for elbow bleeds or shoulder bleeds. And then my mischievous looking other cousin Herb, right down in the corner, and Herb is also sitting beside me down at that table here today. So there we are back in 1964. Now not shown is my mom's twin sister, who of course is the mother of Herbie and Tommy. Now at that time, the factor replacement therapy in those days was 2 pints of plasma. And it was great to ask Dr. Davie, did you really understand at the time how the plasma was stopping the bleeding? And he said, no. And they really didn't at that time. So they just gave it to us and it appeared to stop the bleeding, but it was rather ineffective. And so it had to be combined with long periods of immobilization, bracing, slings, and we really were in the hospital for a long, long time. Now, because time is limited, to be really short, to show the impact of the research and the advancements in care, there I am just about to enter grade 5. A few months later, they decided to put me in a wheelchair for 2 years to immobilize me. So all the time outside I was in a wheelchair, all the time at school in a wheelchair for 2 years. I was prohibited or banned from taking physical education in those days all the way to grade 12, and that was 1972. And so I went from that type of life to, in 1979, graduating the top of the class in physical education at the University of British Columbia. Well, there. My ego satisfaction goals have been satisfied for today. If you don't believe, there's my name up there, 1979, and the picture really doesn't appear on the plaque, but I wish it did. So that's how far we came. So how did we get there? And really, I can joke about that. I can be jocular about that. And I'm not trying to be, well, I have a hard time being falsely modest anyway. but I won't be, because it is a remarkable achievement. And the reason why I can joke about it and present it and boast about it unashamedly is because when they roll out the list of credits for that achievement, I'm far down the list. I don't even think the font size for my name will be the same size as those above me. So who are they and what are they and how did I get to that state? Now first, at the risk of sounding like a football player after a victory, I have to acknowledge the unsearchable thoughts and grace of God. Because, let me give you one prime example. At any single time in the 50s and the 60s, I could have had a target joint on my left knee. I forgot to brag about my left knee. It's beautiful. It bends beautifully. It's right up there. It is a gorgeous knee that never had a major bleed in it. At any time in the 50s or the 60s, just a twist or two to three times, and then the vicious cycle of bleeding would have targeted that joint. And I don't even enter the door of a gymnasium if my left knee is as bad as my right knee. So I think I popped up a quick slide. These brothers from Montreal in my era, you can see if you have two knees affected, it's really very, very damaging. I also, of course, have to acknowledge that beautiful woman in the background and her beauty. My mother, her strength of character exceeds whatever you see there. She was a gorgeous, devoted mother and was great. So those are the two primary credits for my achievement. But the ones that are of particular interest here today, I believe, I regard as the three pillars of hemophilia care. Now, first, of course, is the medical research. It doesn't happen without the researchers in the crowd. It wouldn't have happened without Dr. Davie and all of his work and all his colleagues. Now, you can see right there cousin Tommy in the sling. Let me give you an example of the impact of research. This is 1964. In February of 65, just a few months later, Tommy was in for a procedure. They were aspirating his knee, which was commonly done. Dr. Jackson showed you a slide of a badly swollen knee, synovitis. Well, that was common in those days, and so he was going to get a knee aspirated. And during the procedure, an NG tube probably bruised his nasal passage up high, and he started to bleed. And they couldn't stop the bleeding. And the bleeding went on for practically 30 days, the better part of a month. They would pack it, he would bleed, they would pack it, and 3 minutes remaining. Okay, we have to really roll here. Okay, 3 minutes remain. So they would pack it, and ultimately they tried this. They tried pig plasma. And at first the headline was, "Pig plasma saves boy's life." And then they kept doing it. They gave him a few repeated transfusions of pig plasma, and his blood pressure soared through the roof, and he died of a brain hemorrhage. Now, had that event happened just 2 years later, with the advent of cryoprecipitate, Tommy's bleeding episode would have been a non-event. Okay, so that's how far we've come. So we're indebted to the medical research. I'm also indebted to the second pillar. Even though we had cryoprecipitate in 1967 here locally, 1968 I was still going to school on crutches and a brace. And the reason being is when we'd have a joint bleed in those days, we'd have to go to the emergency department. We would wait hours for the transfusion, and even a minor bleed was a significant joint bleed by the time we were done. So the cryoprecipitate didn't change my life entirely. It wasn't until we had a clinic here in Vancouver, and clinic care and the comprehensive team, they moved the transfusion right into the home. So they taught us to self-infuse, and at that moment, my life was changed. It was 1972, and no longer did we have to wait to infuse. So we could infuse immediately and very, very quickly, and it changed my life. Now, the last thing that today's hemophiliacs tend to lose out on is the peer support. And we all knew each other in those days. We were hospitalized together. And of course, I had my cousin Herb. And I had started a business career. Cousin Herb, whoops. Cousin Herb with those eyes. He says, "Tim, I'm going into physical education." I said, "You can't do that. You're a hemo." And I'm not sure what he said, but you can imagine. And so he showed it could be done. Life took him in a different direction. And I really liked what he did. And so within a few years, I dropped my budding business career, and I followed suit into physical education. But I wouldn't have done that without his groundbreaking procedures. Also, his groundbreaking procedures, sometimes it's really important to listen to patients. We were taught, when we were being taught to self-infuse, rotate the vein sites. That'll save the veins. Herb would say, no, Tim, go through the same spot every single time, right through the bruise, and you'll get a nice little scar tissue tunnel, right, a perfect portal into the vein. Well, this baby has been working beautifully for 40 plus years. It was against all conventional wisdom. And and that really is significant for young hemophiliacs and young parents to make the infusion go much more rapidly. So the peer support, so Herb and I knew at that time, and I know my time is waning, so I'll try to be quick. We knew that if we could pack a joint at that time with muscle support and proper strenuous physical activity, you're not going to bleed. I stop bleeds in my elbows by getting strong forearms, and you're not going to bleed. So that was our philosophy. Now, it did have to slow somewhat in the 80s. The blood problems, the tainted blood, we had to back off from that because we wanted aggressive treatment. I used a lot of cryoparticipate when I was in phys ed, but we had to back off because of the blood. And so the message isn't quite as strong today. So my life has been hugely impacted by the medical research, the clinic care, and the peer support. I can't take you through the 80s. I'd like to. I was one of the few hemophiliacs who missed HIV. I did get hepatitis C. I did get colon cancer in the 80s. My life was saved by early bleeding from hemophilia, so I'm very happy to be a hemophiliac. I was able to clear the hep C, and today I'm one of the patients of Shannon's that's on the daily infusions. And that really has been terrific. The daily infusions, even better than the 3 times a week that I was on. All joint pain, it reduces it. I play men's doubles tennis 3 to 4 times a week. I don't even think about a bleed. It just doesn't happen. And they're quite vigorous games. And so I'm a very grateful man to be standing before you. And I'm grateful for my tennis games. I do have to give one more piece of credit here, and that's to my brother Mike, because he said, make sure you give me credit, Tim, because I couldn't play tennis 3 to 4 times a week, first of all, without a good anti-inflammatory. I hope it's safe. And without the help of being ambidextrous. And my brother gets the credit for that. He's not affected with hemophilia, but at a very young age, he decided that he was going to make me a left-hander. And so he honestly tied my right hand behind my back, and I threw left-handed, bought me a glove for my right hand, so I really did throw left-handed till I was about 12. And then every time I threw left-handed, I'd have a joint bleed. And so I just reverted back to my natural right-handed, and now tennis I have 2 forehands, and so it's really quite helpful. Now, good, I haven't seen the lady with the clock just yet, but 9:53, I get till 10, you don't want any questions, just a little bit more, because I really have to take a little time. It's such a great opportunity for me. 2 minutes to thank the researchers in the crowd and the medical community in the crowd. I think sometimes you in the medical research field become disconnected from the impact you have on lives. And you have a hard time getting through your bureaucracies and your appeals for finances. Yet your committed efforts really does change our lives. Herb and I, those days, I could go on. We really did have a lot of pain, and I could cite many instances of the great pain that we had. But when I compare myself, not graduating the top of the class, really, I didn't even know that award existed. So I never worked at it. But I worked at artistic gymnastics. And I can still picture myself getting the highest mark on the parallel bars and the best front uprise on the rings. And whenever I think of that image, I contrast it to the pain we had as young children, and another layer of deep gratitude is laid in my heart. And if that image wasn't enough for you, I have to finish with Herb, 'cause this is now Herb's time. Now, there is my competition. There is my competition and my inspiration. Now, see, he's the one who led me into phys ed. Look, I'll zoom in a little bit. Look at that face. Look at that face. Now, he's not skiing just on one ski. He's skiing on one leg because he's got a little stick of a right leg that doesn't bend at all. You can see it tucked in there on that turn. On the left turn, it would flutter in the wind. His little twig of a left elbow, that's not really holding on because he couldn't move it. It was total flexion deformity there. And you might see this massive right arm on the other side. And that's what you can do with hemophilia when the factor replacement was so poor in those days. Our hope and our goal is that every young hemophiliac today has both arms like Herb's right arm, has both legs like his left leg. And it's an honor to be speaking again before you. I just wanted to give one quote from my researchers. It's a quote from Solomon, and it's old, but it goes something like this. It says, "It's the glory of God to conceal a thing, but the honor of kings to seek it out." So I thank you all for striving continuously to reveal that which is not known. Thank you.
What advice do you have for hemophilia treaters on how to approach teenage boys
You know,
who don't want to do their infusions?
That's a great question. Shannon asked me about that for the young boys. I understand they're still having problems with their ankles. I can't come up with much. It's the team approach. There's medical research, there's the caregivers, and there's peer support. I personally would say, hey, if you're having trouble with your ankle, here's a beautiful ankle cast that I have, the air cast. They work beautifully from my experience. But I was thinking for young boys, not all young hemophiliacs are sports-minded like Herb and I were. And I think, wouldn't it be great for the clinics to have a clinical psychologist on board to do psychological profiles, specializing in motivation and behavior, so that the clinic staff could always access that information to see what turns the crank of each individual hemophiliac. Great. Thank you.
Okay. And now we're on time. So our next speaker is Dr. Valter Arruda, and I'll start off by apologizing for leaving him. I invite the speaker from University of Pennsylvania, comes up, said we'll meet for dinner with the group. I leave him without taking him to dinner. So I owe you a dinner, and that's not generally how I treat most guests. But that's not really the introduction. The introduction really is that Dr. Arruda is an associate professor of medicine at the University of Pennsylvania, hematologist at Children's Hospital in Philadelphia. He completed his medical training in hematology oncology training, as well as a PhD in molecular biology in Brazil, after which he did postdoc fellowships in Philadelphia and then in Leiden. And then finally returned to Philadelphia almost 20 years ago, I guess, where he's really been a leader in developing gene therapy approaches for hemophilia A and hemophilia B. And here you have a great example, as we've heard this morning actually as well, is somebody who brings together the science and the clinical aspects together to solve what are urgently needed problems. And you can see here now today how exciting it will be when we can physicians such as Dr. Ruda can treat patients with hemophilia with hopefully a one treatment, do all, and never again kind of approach. So.
Well, first of all, I really like to thank the organizers to give me opportunity to go ahead and try to discuss the question that Ed gave me, what is the future and obstacles of hemophilia treatment using gene therapy. But I want just to make a point here, there's the theme of 1964. I just figured out now that I was born 2 months later that the cascade was described. So pretty much close to my heart, I guess. So what we've been doing over the last almost 20 years is trying to simplify the treatment to hemophilia, which I don't have to remind you what it's about because my previous colleague said. But at the bottom, you see that we are a little bit like pretentious. We want to say that gene therapy should be once and done. So what we want to do with this is actually take advantage of what we know from the clinical phenotype of the hemophilia that even modest elevation above 1% might be enough, or in some cases, sufficient to change the disease phenotype, such as 1 to 5% of the normal be associated with the moderate disease. And now we're going to see at the end of this presentation that we might be able to aim at higher within the mild range. And basically what we want is to change the baseline, provide a simplified prophylaxis that will prevent most of the spontaneous bleeding. Of course, depending how much the patient exposes himself to trauma, that might not be enough, but overall should be okay. So, and therefore, we want to prevent most of the bleeding, avoid the frequent intravenous injection of clotting factors, and as discussed before, nowadays is much less of a problem, but we only test for things that we know exist. So there is always the concern that might come up something different that we don't know in the future in terms of blood-borne infectious disease. It's particularly in the pediatric, no need to the central vein, which is given 2 ways, access to the treatment, but also 25% of people are complicated by either infectious or thrombotic problems into the central lines. So although hemophilia A and B, they are clinically identical, we know exactly the therapeutic range, that's where the similarity stops when it comes to gene therapy. From a gene therapy perspective, these are very different diseases. And here I highlight what they are. Factor VIII cDNA that need to be packed, as well as the factor IX, is threefold larger than factor IX. And that's explained why historically most of the trials start with the factor IX gene therapy, although only 20% of the cases in hemophilia are doing hemophilia B. But we get better over time, and now we can actually effectively package and express good levels of factor VIII in some systems. Also, it's very well known, not only for gene therapy, but as well as for recombinant production. Factor VIII is notoriously an effective express, and the purification over the years proved to be quite a challenge. And this is true not only for in vitro, but also for in vivo. And there is a potential risk of formation of antibodies to factor VIII or factor IX as any other novel gene therapy for hemophilia. Gene therapy won't be exception. Therefore, the risk is much higher for hemophilia A than for the hemophilia B. Although we always knew that the therapeutic levels in terms of concentration for factor VIII is much lower than for factor IX, actually even to express comparable activity levels is notoriously difficult in different systems that we learned over the years using either small or large animal models of this disease. So what we are nowadays is what I'm going to focus is, there is a variety of approaches, but I'm concentrating ongoing and some planned clinical trials that actually over the years, the last 10 years, mostly after the last 5 years, converged to a single vector class. And this is using a vector based on the virus called adeno-associated viral, AAV. And here's just to remember, this is not an adenovirus. AAV actually is a parvovirus belongs to the parvovirus families, is a replication defective and non-pathogenic. Has a limited capacity, that's why I explained it was hard to work at the beginning with factor 8, which is 4.7 kb. An AAV vector is made by removal of the happy cap genes and replace that with your therapeutic transgene, your interest with promoter and regulatory regions. This we call, based on the parental viral, now we have the recombinant viral or vector. So AAV vectors, as you can see, the reference like old, but over the last 20 years we learned that AAV is associated with both long-term expression and sustained levels of therapeutic levels of factor 8 or factor 9 in not only in small but large immunocompetent animals. And here's just to give an example, when we clone the canine factor 9 in front of a liver-specific promoter that restricts expression to the hepatocytes and was delivered to dogs who has severe hemophilia B. This is the dose that we use, 1 times 10^12, and the unit is vector genome per kilo. At day 0 of the protocol, 3 dogs were treated with this vector, and we follow them for a long period. As you can see here, after a single injection, over more than 2 years, the levels range in around 6 to 8% of the normal. So therefore, this can be associated with a mild phenotype. And the safety profile in several different models, animal models, were actually quite excellent. It moves that to initiation of clinical trials. Because we do not sacrifice these fragile, very important dogs, we follow and ask the question how long the expression lasts. These are the 2 dogs that we follow over 8 to 14 years, and after a single injection, the levels of factor 9 that was achieved more or less after 6 to 8 weeks following the IV injection of the vector is still stable over more than a decade. So that's what we want to do with the clinical trials in humans. This is published back 10 years ago, and these are the 2 patients enrolled in clinical trials. In the first figure to the top is the patient that expressed received 2 times 10^12 vector genome per kilo of the vector very similar to the dog, of course, expressed the human. And here what happened after vector administration, this patient expressed levels around 12% of the normal. And this, I remember, was the very happiest weeks of my life until we start to learn this thing. Contrast to the dogs, the factor 9 levels start to drop. And this coincides to increase of 2 markers we call ALT, AST, that these are liver enzymes. So there was a liver toxicity that after Cs, the factor 9 returned to the baseline. We have several discussions that the FDA requested us to do a second subject with a lower vector dose, here 4 times 10^11 vector genome per kilo, and we knew that at this dose we should not have factor 9 expression, and we didn't, but we saw a transient increase of this liver enzyme very similar to that. But if you look here, the unit is 600, this is 150. There is a nice correlation between the vector dose and the liver toxicity. Therefore, this approach suggests that there is a toxicity mediated by the vector capsid, and this is vector dose dependent. And none of these patients develop ever an antibody against the human factor 9. This also shows us that these liver enzymes can actually be used as a good biomarker. And this is not our general training, which say 2 times above the normal. This is 2 times or 1 time or 1.5 above the baseline. Even levels that we do not care too much, 20 units, if this goes like 40, 50, is a red flag. So it's a good way, very sensitive biomarker that can be used. And the reason for that is after 5 years of intensive attempted several groups, including ours, we could not come up with a preclinical model that we can test this capsid-mediated immune response. So only clinical trials will be. The reasons why only happen in humans we can discuss, but basically after 5 years we discuss with the regulatory agency and we move ahead. But with the one thing, we think that should be a vector dose should be monitored closely. So these were the lessons that we learned for the early trials. Intravascular delivery is the the capsid, as I mentioned to you, but also we learned if you have neutralized antibodies to the AAV capsid, that will prevent effectively transduction in vivo. So based on that, there was also a boom in the field of what we call alternate serotype. The serotype that I showed before, it was the very first to be tried in humans, is AAV2. But then we learned that there are vectors that have a much different serotype, they have a higher tropism for the liver. This simplifies, number one, the treatment, because the first we have to do into the hepatic artery. By using vectors, the higher tropism for the liver, you can just do a simple peripheral IV injection. And the vectors that I'll talk today is more based on the AAV8 or similar to that, and AAV5. And what we learned over the years is that overall, for each one of the serotypes, at least 40% of the population, sometimes even more, will be test positive. So we need alternative serotypes because one serotype won't be eligible for all patients. So these are the complete and ongoing clinical trials. Here I highlight what the sponsors, academics, and biotech companies. This trial was the lead one in terms of follow, and now it's closed. They differ, in terms of the serotype used, could be AAV8 or very similar to that with comparable liver tropism, AAV5. They are making different systems. This is making mammalian system. The AAV5 is making insect cell lines using a baculum system for transduction. And the transgene here ranges for the wild type factor 9, a variant with increased specific activity that I'll talk later, factor 9 Padua, as well as we have the very first trial using factor AAV5 for expression of the B domain deleted human factor 8. So the group from St. Jude's, together with the University College of London, they published two papers, one in 2011 and the long-term follow-up in 2014, showing intravenous injection of AAV serotype 8 express wild-type factor 9. And the data they show, when they went to the high-dose cohort, which is very similar to the AAV2, 2 times 10^12 vector genome per kilo, they saw this curve here. Again, factor 9 is showing red. Initially, this is transfusion. The expression was going okay until they start to see a drop and barely increase the liver enzyme. They start to go and use a transient immunosuppressive prednisone. So they were able to rescue this transduction rather than what we learned in the past that we didn't do anything and we lost the expression. And here is just, you can take PBMCs and look at the and put them in culture with the AAV capsid peptide library. And you can see there is a nice correlation between the drop of factor 9, a little bit increase of liver enzyme, and expansion of this pooled memory T cells that actually are mediating a cellular immune response. But upon install of the prednisone, this actually contracted significantly, which was a sort of surprise. And together, in terms of efficacy, these are the whole group of patients. They have 3-dose cohorts, and these are specific for the high-dose cohort. As we heard before from Dr. Shannon, it is interesting because one subject that actually was the mid-dose cohort expressed 3%, which could be good in general terms, but was a 62-year-old man with a really severe joint disease, which didn't actually help too much, the 3%. So he still needed prophylaxis. Not at the same amount, but it's still going on. So this we will learn over the years how better tailor which patient should be aware or based on the strategy that you have. But in the high-dose cohort, it's clearly that you reduce the consumption of factor 9 as well as the rate of annual bleeds significantly reduced. So I talked with Andy Davidov with his PR St. Jude's before I visited you, and he updated me on where we are nowadays. The high-dose cohort is 2 times 10^12 vector genome per kilo. They have a total of 6 subjects, and 4 of them require steroids. What happened in terms of efficacy? In total, there are 10 patients. The levels are vary from 2 to 8%, and the follow-up, the minimum is 4 to 6.5. And according to Andy, after 6 months, the levels that they were achieved sustained over the period. So it seems to be like they are doing what we saw initially in the dogs. The immune response to the AAV capsid, the cellular immune response, was restricted to the higher dose cohort, which tells us that probably this is the B dose cohort, 6 times 10^11 per kilo, might be a threshold that below that you're safe. Above that, you have a risk. So the question that we ask is, can we lower the therapeutic vector dose? There are, serious work trying to find out more alternatives to AAV serotype that probably have a higher tropism for the liver, therefore you need less vector. Or we can use a common approach, which is called optimization, which is in the gene therapy, like everybody does this. And we also try to look at hyperactive clotting factors that might be able to do it. So we are happy to have collaboration with Paolo Simeoni from the University of Padua in Italy, that he actually identified a young man with a unremarkable deep venous thrombosis. What was remarkable was young, but no family history and no risk effect for inherited thrombophilia as we do usually in clinics. But what was a surprise is that this young man has a factor IX activity over 700% of the normal. But the antigen levels are normal, so you can calculate the rates of activity to antigen was around 8. He has a young brother like him as well, the same rate. One brother completely normal like his father, and his mother was intermediate, which clearly suggests a X-link. Inheritance, we find out that arginine 338 was changed to a leucine, and this was associated with an increased protein-specific activity. In the lab, we call this a Robin Hood strategy, calcium. Can we do this to lower the vector dose that we can? So these will be purified proteins on top, just show that the recombinant protein express the same increased specific activity as well as if you activate, which means that it's not activated in a different way from the zymogen, which is a good thing. So we spent a couple years using inhibitor-prone hemophilia B dogs that have an early stop codon and are very sensitive to development antibody. And actually the dose shown here, 1 to 3 times 10^12 vector genome per kilo. We get supraphysiological levels of factor IX in some of these dogs, like 200. But importantly, the specific activity was very comparable to what we see in the human family. So there is no immune response to the factor IX Padua variant. Continuous expression by AAV eradicates an antibody in one dog. This is published, I'm not going to show you. But important, all these dogs follow for a very long period of time, show no evidence of thrombosis. So these form the base for 2 trials that are ongoing, and one of them probably stopped, I don't know. But they were using the variant factor IX Padua, and these are the serotypes, either AAV8 or Spark 100. Your guess is good as mine. I just know that this works like as good as AAV8. So this is the data from Spark. What they did, they used 5 times 10^12 vector genome per kilo. This is below the 611 that we knew that was safe in the early trial for the AAV8. So here is the follow-up of 12 weeks of this first set subject. This is the injection at the beginning. All of them are now following for very long period of times at the range of 20 to 35% of the normal. The only one, the short follow-up is subject 7, that here a few, 2 weeks is around 15% of the normal. And we learned that this patient not only has sustained therapeutic levels, which all of them in the range of the mild hemophilia, and some of them actually very close to the 40. These blue bars are the infusion of prophylaxis or on demand. And after gene therapy in these 4 initial patients, there is no treatment. Only one subject received this. We'll learn more about this phenotype a few weeks from now during ASH that will be presented by Dr. George. But what we learned in the last week is one of the 7 subjects, actually subject 7, developed a AAV-mediated immunocapsid response that was rescued by prednisone, as I showed you before. So even if you lower the vector dose, it's still a risk that some of these patients will. But the difference here is 1 out of 7 rather than 4 out of 6. So it seems to be a good improvement. The vector dose reduction is feasible, but other factors may actually trigger the immune response. Oh, okay. So, and together with the data that we call now SHIRE, there are a total of 15 subjects that express factor IX Padua. There is no evidence that this actually is associated with the formation of inhibitor. So change gears, I will show you this just one trial that was done by Unicure. They use AAV serotype 5 expressing factor IX wild type, and this is made in the insect cell lines. I'll just mention this because probably there is intrinsic problems, no problem, but specific characteristics of this vector. These are those that they decide to use, 5 times 10^12 vector genome per kilo, which is almost 3 times what we learned before that was associated with immune response. But actually, they couldn't find any probability. Only one subject out of these 5 actually has a transient elevation that took very long time to show up, around 10 weeks, and was fine. And overall seems to be effective. There is a reduction of the annual bleeding rates, but one of these 5 patients remains a prophylaxis. But this is a quite high dose. But this formed the base for the second trial that was actually used for the only trial that we have for hemophilia A, which is using AAV5, a codon-optimized human factor VIII B domain deleter, made in the similar system. And this was done by Myron. They showed this data in July for the World Federation of Hemophilia meeting. And here's the results. They start, surprise or not, to 6 times 10^12 vector genome per kilo. How they did it, I don't know. Just like very brave. And the idea was, after 4 weeks, if the patient's not expressing 5% or higher, they will go to the next cohort. So that's explaining we have one subject in the low dose, if you will, mid dose, express only 2%, and then they start the high dose, 6 times 10 to 13 vector genome per kilo. All of them expressing now over 10% of the normal. Some of these patients have actually supraphysiological levels. As you expect, there is a substantial reduction of the unknown bleeding rates in 6 out of the 7 patients. But here's what's, if you look at from this, I took from the website, they don't give me anything, I just found this. It's the subject 3, is the first one in the high dose cohort. In contrast with most of the clinical and preclinical models, plateau is achieved after 6 to 8 weeks and remain there if you don't lose expression. This is quite unusual. There is a long increase of the liver of the factor 8 levels here in blue, and also increase the liver enzyme. But there is never a loss of expression. Because this takes the liver enzyme start to increase at week 4, all the other patients, 4 to 7, they did prophylactically. They did not expect the liver enzyme to go up. They just installed the prophylaxis right away. And this is what you can do. You can normally look at the 7 patients over time. The factor 8 levels actually went all the way beyond the normal levels, the actually supraphysiological levels. And I don't have any more information on that. But the outcomes of this show good. Is no immune response to factor 8. Subject 3, as I showed, start to increase the liver enzyme and set the pace to all the other patients. They will receive the same way, and there is no loss of expression of factor 8 despite the increased liver enzyme. It's unusual kinetic expression, which I'm not familiar with. The great concern about the supraphysiological levels, no clear dose response, and seems to have an ongoing liver toxicity that we learn. But in summary, we have 35 subjects now with hemophilia B and A. They're enrolled in distinct clinical trial, distinct serotype. All of those express more than 2% of the endogenous factor 8 or factor 9, show improvement in the disease phenotype. Is dependent on how the joint disease were. Some of the strategies are more predictable of the outcome and with consistent response. The AAV capsid-mediated T-cell response and loss expression is vector dose dependent, also serotype dependent, and the onset range for 4 to 10 weeks. There is no sustained acute or chronic toxicity and no formation of antibody to any of these transgene. In conclusion, AAV-mediated strategies to achieve therapeutic levels of factor 9 are feasible and safe. Early data for factor 8 to be are in the same ballpark, but we need to look at the safety profile that will tell us after all the subjects stop steroids and how the levels will be. The main obstacles that remain is actually the bottleneck effect is the presence of neutralizing antibodies to the AAV capsid. So as I said, 40% of people will be excluded, so the idea is to select only people who are completely naive or negative, attempt to develop alternate serotypes. This is ongoing. Immunosuppression plus is actually they have very poor outcome. And the cellular immune response, the only reasonable thing to think about is the lower the vector dose, but depend on the serotype and as well as the transgene. There are serious concerns that we don't know yet. We can compare side-by-side vectors made in different systems, in different purifications, and we like to learn a little bit more, as well as to learn what is the liver enzyme underneath this AAV5 for a factor 8 trial. An alternative, I think, would be to develop factor 8 strategies that can be optimized with expression and make the vector dose not only this only trials using a unique system. I think for the other system based on factor 9, we're not going to get this supraphysiological level factor 8. And one place to go, this is just to summarize, I think the B-domain delete is quite forgiving in terms of immune response, and this is just published. I'm not going to show you this. And the second thing is if the supraphysiological levels actually remain a problem, we might need to develop a regulatory system that you can turn on and off. But I don't know. I thought, to be honest, that in my lifetime I will never have to concern about supraphysiological levels, much less a factor 8. But consider what happened the last 2 weeks, I think how much can we really predict things. So I'll stop here. Thank you.
So, Valder, I'm not sure that I heard it correctly, but the Shire trial, which is very similar to Spark, is not getting the same results that
That's
the
correct.
Spark trial is getting. And they're losing expression, but without the transaminase elevation. Is that correct? And do you have a speculation why that is?
Yeah, well, I think these are different vectors in terms of the capsid. The one is AAV8, the other one is derived from the AAV8 likely, but there are a lot of nuances how you purify, and because you can have the same vector expressed in the same cell line, it's depending how you purify. It means how much of the empty capsid is left and how much you can correlate that. Technically, it's stated that around 10% of the vector prep has empty capsid, which is different from the other trial that seems like they have no empty capsid, at least made in the vector. So, but the other thing is these assays, Nigel, is quite in-house, very difficult to compare not only how tight your vector, there is no normatization for a Qantas lab, much less in terms of how you detect neutralized antibodies and how we follow the T-cell capsid. So what we learned from, as much as I know, what Paul Monahan, they have one subject in a cohort of 1 times 10 to 12 per kilo that actually is expressing around 20% over now 28 weeks or longer than that. So, but that was not consistent. So why that trial did not reproduce? I mean, they do have a peak of expression, they lose quickly. I don't know. What we learned from, I speculate, that what we learned from the clinical trial from London, when the very first patient, the high-dose cohort came, was a Friday. So they saw a blip of the liver enzyme, not much in the factor 9. So they say, okay, come back on Monday. When they got there, they lost 50% expression. It's depending on how the system was set up. If you wait more than 48 hours, the chance you're going to lose is very high. Because you're making factor 9 powder, you express amount, but the amount of cells are pretty much the same. So you lose as much as you gain. So that is my speculation. I think it was how, if you interview, it doesn't matter the PBMCs. The PBMCs is never real time. You can only look back and try to put together. So I don't know. But I know that they stopped the trial, they are redesigned.
Valder, are you aware of any strategies for factor 8 that are similar for what you've used for Padua? For instance, maybe that disulfide stabilized 8 that I think, was it Stephen Pipe used or made a while ago?
Oh, is it? No, it's a different group, but what I know in terms of trying to improve the specific activity of factor 8, the best what you have is a chimeric, which is the porcine in human, but there are intrinsic problems with that, but I'm not aware of that. And in the lab, we are trying to mirror or try to understand how 9A and 8A interact and try to mirror with the same thing towards the factor 8, if that can recapitulate the part about from the factor 8 side. We don't have any result yet, so, but we are going in that direction.
Thank you.
Okay, two quick questions. One is FDA trials. The minute you see a blip in liver enzymes, you get into problems, and quite a lot of trials have been actually preempted by raised liver enzymes with no longitudinal evidence of cirrhosis. So one issue, obviously, is what do the liver enzymes actually mean in terms of long-term prognosis? It'd be silly to throw out a good treatment for something when you don't actually know the outcome. Difficult to study. The other thing is this immunomodulation. There's a lot of interest in the oncology field of taking out patient cells, modifying them, putting them back in, rather than just going with something like steroids. Is there any sort of movement in this direction, or is it too soon?
Well, I think there are several. I'm going to make comments. I hope I'll address your question. When we saw first this immune response, the consensus was there was a pool of memory T cells that were expanding. And we talked with hepatologists and nephrologists in the context of the organ transplant. They thought these are very difficult cells to get rid of. So we were expecting our group at Penn using a combined MMF and rapamycin. So, and by surprise, we caught by just using straight prednisone, was sufficient. So, show how much we knew about it. So, I think that's the point. The second thing is, I think the field is moving towards preemptively, like this design from the biomarine trial. One patient, the specific dose cohort, show liver enzyme increase every week 4. All the other patients should do preemptively. Will this be the most effective way? Could be. And I think there are other trials coming the same design, and we will learn that. So you just prevent rather than go after the damage. When we have the very first subject back, over 10 years ago, we have a serious discussion with pathologists and GI people, and the idea was that there is no way to predict the extent of the liver enzyme based on the amount of the ALT or ST. So there is no correlation there. This was based on when you have drug toxicity, that seems like there is no such correlation. And we were asking them whether we should do biopsy, stuff like this. They said this will be useless. So that's where we are. And the FDA, what you propose, as I said, there is no preclinical models that we can address that. You have to come up with a vector dose that probably will be in the ballpark to be therapeutic. But I really don't, is a difficult call there. That's what I say. I was surprised, probably many people were, because these two trials, they use very large doses. They are based on AAV5. And there are two problems here. One, AAV5 is not very good for liver. But what they want to do this, because in the general population is the lowest prevalence of people with neutralized antibodies to the AAV is towards AAV5. So you have a lot more people that you can enroll. But because it's not that efficacious, you have to deliver a higher dose. It seems like the toxicity that we found with one serotype does not apply to the other. So each model you have to go probably doing clinical trial very slowly, very carefully to see if you can move ahead. I think that's the only way to do it. Did I answer your question? Okay.
Thank you very much.
Okay.